The overall goal of this procedure is to determine the effects of various agonists, antagonists, or novel agents on ex vivo human myometrial contractions, to investigate myometrial physiology and to validate pharmacological data from novel research probes or drug leads.
This method can help answer key questions in the field of myometrial physiology and pathophysiology, including preterm birth and dysfunctional labour. The main advantage of this technique is that once mastered, it's a relatively simple technique that can provide useful information about the functional responses of novel compounds in vivo.
Before beginning the procedure, position one peristaltic feeder tube per tissue bath around the rollers of the peristaltic pump head. Secure the tubes with the retaining stops by tightening the compression cams and locking the keys around the tubes. Place the free ends of the peristaltic feeder tubes into a 1-liter container of physiological saline, or PSS, and start the pump to allow the PSS to continually perfuse into the tissue baths. Place the pregnant human myometrium biopsy sample into a clear celastic-based dissection dish filled with PSS and place the dish under the microscope. Carefully orientate the biopsy so that the serosa and decidua edges can be visualized. Secure the tissue to the dish base with dissection pins. Using the 10x objective, identify the regions of myometrium free of scar tissue, serosa, decidua, and adherent fetal membranes. Then, use large